Hello, everybody. My name is Anthony Davies. I'm the Chief Executive of Dark Horse Consulting Group. It's my pleasure to introduce to you virtually a cell and gene therapy meeting on the med virtual panel discussion. You can tell it's virtual because of the variety of fake and real backgrounds behind us, and you can tell that we're actually not meeting on the Mediterranean. I'm genuinely regretful about this, and it's my sincere hope this is the last time we have to do it this way. I would like 2020 to be remembered not as the year of the pandemic, but as the year that vaccinology changed forever, and probably for the first time in 50 years since perhaps the Salk vaccine rolled out in the 1950s. Gene therapy in particular was instrumental in affecting this change, and I think we all feel very proud and happy about that, that gene therapy to a certain extent saved the world from a much worse fate. Today, we are going to discuss the subject of CMC, chemistry, manufacturing, and controls. Yes, we still use that antiquated abbreviation for all things manufacturing in the cell and gene therapy world, as well as in the rest of the field. And the question we're going to be discussing is, are we moving in the right direction? So it's my pleasure today to introduce a panel of 4 distinguished specialists in this field. And I'm going to go around the virtual table here and ask them to give us a quick introduction to themselves. And I'd also like them briefly at the end of that introduction to reflect on how, when, and why they got into this field of cell and gene therapy. First of all, it's a pleasure for me to introduce Rupa Pike. Rupert is the Director of Enterprise Science and Innovation Partnerships at Thermo Fisher Scientific, and also has global corporate accounts responsibility. Rupert, welcome to the discussion.
Thank you so much, Anthony. Hello to all the other speakers. Happy to be here. As Anthony said, I also hope this is the last time we have to meet on Zoom or any other virtual modality. I'd prefer to meet in person as well at some point in the future. I have always been part of regenerative medicine. I trained with Dr. Jamie Thompson for embryonic stem cells and iPS cells a long time ago. So it was a very easy and gradual transition for me to get into regenerative medicine and then cell therapy. And I've worn many different hats in the cell and gene therapy field, including heading operations, MSAT, technology transfer, process development, and analytical assay development. I'm very passionate about cell therapy because I always think about the patient who is at the end of this therapy. So thank you, Anthony.
Thank you, Rupa, and it's nice to hear somebody, having worked at Geron for half a dozen years, it's really nice to see somebody from Jamie's lab still persisting in the field. Stephen Ward from the Catapult. Stephen's been the Chief Operating Officer at the Catapult and held many other roles one hears over the years, perhaps more off the record than on the record, and really held a lot of the glue together at their now world-renowned Stevenage facility. Stephen, pleasure to have you here.
Thank you, Anthony, very generous. Yeah, and I'm delighted to be here, actually. Much better on the med. The cocktails will be much better there than at home, that's for sure. So as Anthony said, I'm part of the cell and gene therapy Catapult from the early days, about 9 years ago. And I've really always focused on that transition between the sort of translational research and making something then robust and scalable. And that's the area that I've worked with pretty much all of my career. Ironically, it did start in vaccines in the 90s. That's where I started. That's where my Ph.D. was in. And then I got into vaccines for chronic infection, which then got me into cancer vaccines in 2001, a UK company called Onivax at the time. So we were pretty early, actually, back in those days of trying to scale technology, how you take live cells and grow them at scale. You know, we were using 160 combined layers of cell factories. It was a wonderful journey back then. I threw quality by design into the mix in the mid 2000s, just to bit of a life raft to try to sort of find our way through this this morass of data and complexity that we were dealing with the cell therapy product. So like Rupa, I absolutely adore this space. I think it's fantastic. Importantly, what we can do for patients and families, it's just transformational, and yeah, it keeps me going every day. I have been involved with some of the UK vaccine work over the last year on the pandemic response as well. As Anthony said, that crossover between gene therapy and vaccine has been absolutely pivotal, and yeah, it's been a privilege to be part of that program as well. So yeah, looking forward to getting into the panel.
Do you miss pure play vaccines? Seeing all this unfold?
No, not really.
You might come back.
It's an interesting diversion, but yeah, I remember in 2001 joining Onivax and coming out of the first sort of week thinking this is absolutely fantastic, and I just haven't looked back.
Yeah. Neil Almstead joins us from PTC, really one of the iconic rare diseases companies in recent history, and rare diseases has a special place in cell and particularly gene therapy. Neil, welcome to the panel.
Thank you very much. I really appreciate being here, and I'd love to tell some of our stories. I think that PTC got into the cell and gene therapy area about 3 years ago with the acquisition of Agilis Biotherapeutics. And with that program, with that acquisition, we did obtain a program AADC, which is a deficiency in the brain caused by the lack of AADC protein, which converts L-DOPA to dopamine. We're very excited by that program, and we would like to see it move forward. It is currently under review with the European Medicine Authority with a filing that we've made, and look forward to the discussion.
Thank you. And also, I'd like to welcome Martha Rook, who's an SVP at Sigilon Therapeutics and head of CMC and analytics. Welcome to the panel, Martha.
Thanks, Anthony. So I come to cell therapy with a background in traditional biologics bioprocessing. Started out my career there at MilliporeSigma, developing new products and new processes for, as I said, more traditional biologics. But fairly early on in my time there, we had a new initiative that at the time we were calling stem cell bioprocessing. So I got fairly early transition from more monoclonal antibodies into cell and gene therapy processing. And then of course, we evolved into viral vectors and gene editing and all of the very exciting novel modalities. And so I feel I've kind of grown up in bioprocessing with the cell and gene therapy space. And one of the things I enjoy the most about it is the new innovation, along with, as many of the other panelists, the impact to patient lives that we have. And I think we'll talk a little bit about it on the panel, but it's one of the beauties and the challenges of cell and gene therapy, that the clinical efficacy is so strong that we always end up playing a bit of catch up on the CMC side. But that's also where you do some of your most exciting work. So I'm looking forward to the discussion with the panelists today around some of these topics.
I think that's absolutely going to be one of the pivotal discussion points today, is the quite remarkable effectiveness of these drugs. I think I've been in cell and gene longer than anyone on this panel, and it really was by pure happenstance. You know, I happened to do graduate studies in scale up of viral vector production at a time, I won't say which decade it was in, but it was a decade when that was an extremely unfashionable thing to work on. And then I happened, a little bit like you, Stephen, to stumble into this field in the next decade, where we were actually manufacturing in the 90s an oncolytic adenovirus at multi-thousand liter scale for a licensure enabling FDA registered phase 3. Then, as you know, the gene therapy field went into the wilderness for a solid decade as a result of the Gelsinger case and other headwinds. That was when I moved over to cell therapies and worked on embryonic stem cell therapies, and we took Jamie's H-line through the FDA for the first time, legendary 80,000 page IND, which is not something I'm proud of, actually. I think brevity was not one of our talents at that point, but the FDA wanted a lot of data. And then it was in 2017, quite a while past Geron and the early days, when the first 3 commercial approvals happened of what I would say is that the current generation of cell and gene therapies. These were data I'd seen mainly from trials like the Eliana trials and the Zuma trials that generated the products called Kymriah and Yescarta. And of course, Lux Turner joined them as gene therapy sort of came roaring back, having moved primarily from adenovirus to adeno-associated virus. And of course, now both fields are thriving. Back in 2017, that year saw 3 commercial approvals. And for a number of years after that, no more than 3 further approvals were seen. The Gottlieb projections at that time when he was commissioner were calling for literally hundreds of INDs and double-digit approvals a year in a relatively short time frame. Frankly, this has not happened. We'll come on to delays and rejections and suspensions in a minute, but it is still a true statement, I'm afraid, that the number of commercial approvals since the golden year of 2017, a year I described, I think, as the year that the field of cell and gene therapy has been saying it's going to have for years. Since that golden year of 2017, in terms of commercial approvals, it's been really disappointing. Why is that? I'm going to go first to the therapeutics developers. Perhaps Neil, you'd like to kick off. You can agree with me or disagree with me, but please show your working and give your reasons.
No, it has been very challenging. I agree with you. I think one of the main concerns that's happened over the past, let's say 4 years since 2017, is increased regulatory scrutiny and a difficulty in manufacturing these cell and gene therapies so that they would meet the regulatory scrutiny that both the FDA as well as the EMA is putting on these products. I think what we're seeing is a tightening of regulations. We're seeing a narrowing of specifications and a more difficulty making these products meet the requirements that the regulatory authorities have put on us. And that in large part is reducing the number of compounds, the number of biologics that are moving forward.
Do you really think that the regulators are tightening up, or do you think it's not more that increasingly products are moving to a late phase in development where regulators have always been pretty tight?
So I actually think you're correct. I think in the early days of cell and gene therapy, there was a little bit of a Wild West view that you could take anything as long as it showed great clinical efficacy, and the regulators would not be very concerned with the CMC package. What we're seeing today is they are quite concerned with the CMC package. They want to have comparable profiles. They want to have data that supports the validation of the product. Just one concurrent validation batch may not be sufficient to get approval. What we're finding, it's more and more difficult for the manufacturers to get all of these batches produced in a reliable fashion, and that the authorities will accept.
Yeah, I think I'll definitely sort of agree with you on that. Martha, do you have a perspective? Is it on balance, are things getting tighter, or are we moving to a place where they're already always been tight?
Well, I think that there is, a growth in the expectations as the field has matured. And I think also that as we're seeing more and more development and indications under development for cell and gene therapy, you're also broadening out the number of indications so that it's not always against a completely unmet medical need. So as you're developing therapies which drive great value, but also have existing treatments on the market, sometimes that bar becomes also a little bit higher and you have a bit more scrutiny. So I think it's a little bit of both. The field is maturing and the regulatory expectations are maturing along with it. And we're also seeing a broader number of targets that are being addressed, which if there is an existing therapeutic available, you have to show the benefit and the risk ratio is appropriate.
Yeah, and I think to Neil's point earlier as well, Martha, we got the gave the recruiting details essentially for skewing away from clinical and towards CMC at the agency. And I think there's a new generation of CMC reviewers coming in at the agency, and to a certain extent, the foundational generation is now moving on. So I think we see some shifts in that regard. And I would say that that change in staffing would probably lean them, if anything, toward conservatism, given that the sort of the breadth of experience is perhaps starting to retire from the FDA. But new, highly technically competent reviewers are being hired, because you can do master's degrees in cell and gene therapy commercialization these days at places like King's College London. From a primarily a services and a raw materials perspective, Rupa, how do you feel about the dismal and depressing number of commercial approvals recently?
Yeah, dismal is the right word, because as you know, there is a lot of excitement, a lot of empowerment for all of the companies, starting from very small biotechs who successfully raised Series A, Series B, had great IP positions, all the way to the giants of biopharma who wanted to get into cell therapy. There was this huge excitement, and you're absolutely right that after 2017, as some people have mentioned in articles, there's a trickle of things. Yes, there is Tecartus and there's Breyanzi, but they're kind of in the same boat. There hasn't been very much. I do agree with Martha and Neil. What they said is that the expectations are higher. And from the FDA perspective, the complexities are also larger. From a CDMO perspective, talking to a variety of different sponsors, what I have noticed is the complexity of manufacturing is high too. Some people want a very potent cell microdose strategy. Some people are trying to understand how to cut down on the number of the days cells stay in culture. Everyone, based on their IP, has now introduced more and more complexities into the manufacturing process itself that makes the reviewer's job difficult to look at the CMC and say, oh, well, here's another angle. Here's one other angle. We didn't think about this. So I think that kind of challenges the reviewers. And then instead of saying something, they will just revert back to saying, need more data, need more clarification in CMCs. And to address what you said, Anthony, about raw materials, there's this thing that we saw a long time ago in biologics and monoclonal antibodies, and Martha can speak to that, the battle between transition from RUO to GMP and what GMP really means, because it means different things to different people. So, people who are trying to use our GMP products, if they can afford it early in process development, have a smoother way to get to feasibility studies, IND enabling studies, and then moving on, versus people who get stuck into the properties of GMP and RUO, reagents, cytokines, media not being the same. So there are multiple aspects of raw material choosing path, multiple aspects of complexities, partial automation, full automation, GMP in a box. All of these things have made the manufacturing process so complex that then it reflects in the CMC, and then it reflects in the reviewer's challenge to address how that CMC looks.
Some of the raw mats, many of the raw mats we use are considerably more complex than most drugs. Viral vectors is an extreme example of this, but media supplements and other materials are, plasmids are more complex than real drugs. Do you feel like we're building something? You feel like you're manufacturing more drugs than most drug companies over at Thermo Fisher?
Say that again. Do we?
I mean, do you feel like so many of the raw materials that you sell are more complex than most drugs? Do you feel like
Sometimes it feels like
you're doing more development work than most drug companies that used to do historically, just to supply?
Most definitely. And there is, I mean, we're trying to take an approach that is flexible, has cost benefits, has a easy transition from RUO to GMP. And we've taken similar paths with ISO 9001 or ISO 13485, trying to help people understand that, having documentation, having QMS systems available and walking the sponsors through all of that is actually very helpful. We also have a lot of people who will say, these RUO materials work very well. There are no counterparts GMP. So then there is a custom option, and it's a good custom option, except costs increase. So there's always that balance between how easily, how more flexibly, and how cost effectively you can transition from RUO to GMP.
Yep.
But yes, there is a lot of work that has gone on into making compatibility between RUO and GMP in terms of product efficacy, product quality attributes, and documentation for it to meet to satisfactorily meet the criteria that the regulators expect.
Yeah, thank you. That's very interesting. Stephen, up in Stevenage, you have multiple inhabitants of the facility who are closing in on some form of licensure. I think many of them are publicly disclosed. How do you feel as the Catapult, and how do you think they feel as they've seen this disappointing rate of commercialization in the last 3 or 4 years?
Yeah, it doesn't surprise me in a way. It disappoints us all, but it doesn't perhaps necessarily surprise me. We've got an industry that's raised best part of 20 billion US. It's high profile. It's impactful for patients. They're expensive. A lot of these treatments. Doesn't surprise me that we're basically, as an industry, saying, yeah, that's great. You're saying you're marvelous. Up your game. So it doesn't sort of surprise me that there's that general, yeah, guys, you need to, be on this a bit more than perhaps where you were historically. I suppose the challenge, Anthony, which sort of feeds into the Stevenage system as well, in a way, is that companies are looking for sort of CMC IP as well as the classic, active modality IP as well. And with that, obviously, comes diversification, as Rupert has said. So, indeed, that's the driver of the sort of Stevenage model, is that companies can come in and they can do their own processing with a sort of a general quality and an operational support. But it allows them to develop that CMC knowledge and processing skill set, because it's important for what they want to do. Of course, if everyone's coming up with a new process, it's a very special process. It's the crown jewels for us as a company. Then, every time you go to an agency, you've got to justify. That's great. You're saying it's different, but if it is different, then you need to do some more characterization, or if you're transferring it to a different site and they haven't seen it before, you need to do more on comparability, because it's a different type of process. So it's a bit of a double-edged sword, I suppose, from that perspective. The upside, though, is, we mentioned this a little bit as a group previously. We don't have a fixed process for a lot of these technologies. So we don't know what good looks like at the moment. We think we're getting closer and closer, but we don't know what good looks like at the moment. So there's still scope to really make some step changes here in CMC. We're not sort of locking into the things perhaps too early. So that's a glimmer of positivity, I think. But we need to start to get to these inflection points. Otherwise, this CMC type of step change which we need, an understanding of our products, and I'm sure we'll get onto analytics and characterization, it's just sort of not there, and people are having to work extremely hard to get something through through a BLA or whatever.
Martha, does it just drive you mad with your background in? I mean, I remember. Let me ask this question a little bit differently. I remember when the concept of a well-characterized biological was, honestly, a little bit heretical, and people doubted whether it was even possible. Nowadays, if you can't describe your biologic in that way, then shame on you. So I guess my question is two. I'd like you to reflect on two things. First of all, how mad does it drive you, the field of cell and gene therapy, from your background, your very diverse background, and especially you running novel therapies at Millipore Sigma before you joined Sigilon, and secondly, well-characterized cell therapy, well-characterized gene therapy. Are we just, I know that sounds a bit ambitious, but are we just falling into the same trap as the monoclonals people fell into 20 years ago and thinking, oh, that's totally impossible, but it's just going to happen eventually? What do you think?
So I think it's a complex question, and with everything, there's multiple kind of pros and cons on how you look at it. So I think that particularly the monoclonal antibody field benefited greatly from their template. And they were able to really, and I would say also in partnership with suppliers, if by and large your NAM manufacturing process is the same template, and there's small adjustments that you make to optimize the well-characterized portion of it, that has a lot of benefits and cost and scale for suppliers being able to have a large enough market size that they can really innovate on products that drive the whole field forward. So there's a real, benefit there. On the flip side, and I think you saw this particularly with monoclonal antibodies and the use of protein A for purification, at a certain point, you're so locked into your template that all innovation by default becomes incremental, because the benefit has to be so huge to get outside of your heavily invested template that the innovation becomes constrained. And I think that the cell and gene therapy space, we have little pockets of modalities that are widely enough used that we're starting to get some of that, not fully templating, but commonality, I guess I would say, in manufacturing processes. So AAV viral vector, very good example. I mean, we're already beginning to see the rise of producer type cell lines, which is something 5 years ago people weren't really, seriously contemplating, and now you're seeing them begin to come in, and that enables, similar on the vendor side and on the supplier side, scale that lets them really devote to innovation that meets the product's need. But at the same time, you have uniqueness that's going to pretty much always be there, and then there's the next generation coming on top of that. So it's encapsulated cell therapies like I'm developing at Sigilon, or we're developing at Sigilon. You have gene editing, which is a whole new modality. So I think we're going to see continued evolution and acceleration of modalities, which is going to help those of us that are really excited by innovation. And at the same time, as you get more companies or kind of scale in the number of people with a particular subtype of modality, you'll drive some of that platformation. So I think it's going to be a mix for us going forward.
Yeah, very interesting. Neil, at a rare diseases company, I don't know how to put this tactfully. You're not going to make so much money off each product. So it's not like you're developing a cure for a solid tumor or something with patient numbers in the tens or even hundreds of thousands. How do you deal with this? Because you're developing the most complex drugs in history to treat some of the smallest, or smaller at least, indications available to you. So the return on investment per product is going to be smaller.
No, it absolutely will. I mean, you're going to have to look at market opportunity. There are diseases which are, let's say, 300 patients. It's going to be almost impossible for any one company to go after that. I do believe that there are groups within the NIH as well as other organizations that are trying to go after those very small indications. Every company has to set a minimum threshold of the number of patients and literally how much you would be able to charge per patient, and that's obviously a very controversial area as well, which I'd rather not get into today. But what I would suggest for all of us, one of the key points: can you come up with, let's say, 1,000 patients, 2,000 patients worldwide that you could treat? That still would be an area that would be of interest to companies like PTC. It is expensive. You do know that you're going to spend quite a bit of money getting the product out there. If you have to run a very large clinical trial, greater than, let's say, 50 patients, it's going to start eating into the number of possible patients that you could treat. And that's also a risk that you do get into. So it comes down to, can you quickly answer your question clinically? Can you have a relatively inexpensive development path forward? And is the product amenable to some larger scale production so that you don't have to make many batches of it? But it is very complicated, and there are products and there's programs that really companies just can't touch.
To Martha's point, to try and drag out something that I think Martha was hinting at, as some product modalities like AAV head towards a more templated process, do you think you might see some synergies from that perspective, where you might have a somewhat generic CMC package spanning multiple indications and products?
I think most companies are working towards, especially with AAV, a templated process. It depends, of course, on the particular AAV. If you're using AAV9 or AAV5, and some companies are very specific, they will only work with one. AAV, like AAV5 is one organization I know very well. That is a way to get a templated process. It is a way to reduce your long-term costs. The purification, the downstream processing is going to be similar for that individual product, and it will basically make it easier then to do your filing as well. So I think that's one approach people are taking. It doesn't always work, and we don't take that approach because we feel that you need to have the right AAV. You can't just say AAV5 is the only thing we work with. It may not work for that disease. So,
and there are some things which will never be templated across indications, perhaps most notably a potency assay, for example.
Yes.
And I would like to use that to move the discussion towards the rejections, the delays, and the suspensions, which this field has seen over the past 2 to 3 years. I'll refer to publicly disclosed information by way of example. Mesoblast received a CRL for a pure play cell therapy. BioMarin had their hematology AAV product rejected. That's probably going to result in about an 18-month delay in resubmission. Iovance has had their BLA submission delayed by at least a year. And each of these examples had analytics as a component. In the case of BioMarin, the analytics were, at least by public disclosure, primarily focused on detection of functional enzymatic restoration in patients and the compatibility of the analytical method used in the early clinical studies with that used in the later clinical studies, which really, again, publicly, they stated they were quite surprised by the FDA's reluctance to consider data from 2 different methods. Iovance's delay appears, again, by public disclosure, to be completely related to the validation status, to the appropriateness of their current potency assay and the validation status of backup assays, which they are developing. And Mesoblast's CMC package in the advisory committee review, which ultimately led to the rejection of their Roma stem cell for pediatric graft versus host, they again received severe criticism, I feel, from the FDA relating to the effectiveness of their potency assay and firstly correlating with clinical effectiveness, and secondly demonstrating batch-to-batch comparability in their scale-up operations. And last time I checked, there's not a whole lot else that a potency assay is expected to do. So failing on those two accounts is pretty bad. Each of you interacts with this analytical issue in different ways. Stephen, you interact with it from a tech transfer perspective, showing comparability, a scaling perspective, whether it's scale up or scale out. Rupa, your company supplies critical materials for these assays. You do assays, you do everything. You supply equipment that does assays, and you have CMOs in your wheelhouse which need to do these assays for their clients. Neil, we've discussed your program a little bit, and Martha, likewise, you've had a foot in each camp at Millipore and now at Sigilon in developing assays. Rupa, I'll come to you first, and really, all I'm asking for from the panel here is your reflections. I really don't mind what direction you go in on this observation that we've made here at Dark Horse, that a majority of the delays, suspensions, and rejections, not all, but a majority of them, I would say even a large majority, track back, at least in part, and sometimes in a large part, to analytical methods, analytical method validation, and more than any other method, to the issue of potency assays. What are you going to do about it? What's Thermo going to do about it, Rupa?
I guess we will need a lot more time to talk about what Thermo is going to do, but I'll try to summarize it. So the way we understand it and the way we're trying to approach and help the sponsors is rooted in three things, right? In cell therapy compared to everything that's established in biologics, monoclonal antibodies. Right now, we feel like there's very limited in-process testing compared to, the gold standards of biologics, because we're always fighting to make the final product, the final number of cells to go in patients. So even though in-process testing is there, I don't think it's as robust as it needs to be. And because of that, we're really not understanding all of the critical quality attributes of the product. Also, I think there is, there could be more. I want to say there's limited product characterization. All of the critical quality attributes are not known, primarily because, it's a closed system going from one unit operation to another, hoping while doing limited in-process testing is the cells are really what they are, they're expanding as we want them, they're getting activated, secreting cytokines, whatever. There's essential elements of product and process related impurities that are not well characterized. And then also the assays are not very robustly developed when it comes to potency, or they're not qualified enough, or they have not been qualified with all of the stability testing taken into consideration. Reference standards are also not either well established, or if they're established, they're not adequately characterized. Comparability, as Martha mentioned, is a big thing. We're dealing with establishing things with healthy donor cells and then applying those same assays to disease cells with patients who have gone through multiple chemos, chemoradiation, combination therapies, comorbidities. So the behavior of the cells is very different. So all of the things that I have outlined, I think are important for sponsors to consider, maybe to take a step and look at each aspect so that when you do come to the final critical quality attributes, that they are sound and they're solid and they're reproducible. And when methods are transferred, scale up happens or scale out happens, we do not lose sight of those critical quality attributes especially potency, because it's so important. So I think that's kind of my, 2-minute take on a very complex situation.
And Martha, you come from a field, originally a much more mature field, where these quality by design metrics like CQAs and CPPs are, lingua franca. Everybody knows them and everybody uses them. The regulators are very comfortable with them. You're coming into a field where, sorry about the traffic, everyone, if you can hear that. There are some people driving on the California roads these days as the pandemic comes to an end. Do you find, let me be honest, do you find ignorance, reluctance, skepticism about CQAs and CPPs and the general, the quality by design approach to things, which, so we say seasoned engineers like you and me know and love?
No, I mean, I think less and less. I would say we had a bit, and I think we're still a bit in the transition where a lot of the, cell processes and some of the gene therapy processes coming out of a more academic development setting and into industrial development, there was a little bit of a lack of that definition. But I think more and more process engineers are coming into the space as the field evolves. And I think that all of us and that those that are now coming maybe more from the biology side are becoming more and more familiar with these critical attributes, and at least are familiar about the language and the necessity and the concept of them. I think that where maybe some of these issues that we're seeing around the delayed approvals are coming from is really a bit what I referenced in the beginning, the speed of development. And so I think there's the beneficial process of setting phase appropriate specifications and assays early in development, and where it's important as developers to think about, okay, from a much earlier point for cell and gene therapies than perhaps people were used to for biologics, what is my comparability plan going to be? How am I going to evolve my assays so that if I get really amazing clinical responsiveness, I'm set up on my CMC and analytics side to be able to take that forward? And I think to a certain extent, it is a bit of a being the victim of our own success that we didn't, as a field, keep up most rapidly enough. And at the same time, we're seeing a little bit of a raising of the regulatory bar as the field progresses.
Yeah, no, I mean, thank you, Martha, for the perfect segue. We all chatted about this yesterday a little bit, didn't we, in the pre-chat? So here's what I think is going on. The panel's heard this diatribe yesterday already, but I'll say it again for the benefit of our audience. And I asked you all to sleep on it, and let's see what you all have to say, having done that last night. I think here's the problem with the field of cell and gene therapy. You're all on the edge of your seat finally to hear what the problem is. The problem is that the drugs are so effective. Okay, these are just great drugs. I remember when I saw the first phase 2 data from Eliana and Zuma readout, and there were effectiveness numbers in the 70 to 90% range, for patients of last resort with liquid tumors, fourth line treatment, and those numbers were, obviously nuts. And they were pretty much borne out in the phase 3. And from what I see of the commercial data, they're still doing just as well. So if a drug can pop out of phase 2 and you can start writing the ticket for licensure, because the clinical data is so great, and this is something we've seen again and again and again, it's going to come out of phase 2 with phase 2 CMC. And neither the FDA nor the EMA, heaven forbid the PMDA, or any other regulator in the world is going to be okay with that. These drugs, furthermore, they're not only coming out of a phase 2 with amazing effectiveness data, but they're coming out of academia, probably with a physician-sponsored phase 1 into a phase 2 without the commercial structure that, PTCs, you've been around for decades developing rare diseases. All of us have been doing this for decades. So suddenly these products are erupting from really commercial grade clinical with barely phase 1-2 grade CMC. I mean, Neil, you've been developing, your company's been developing drugs for rare diseases for much longer than this has been happening with cell and gene therapy. This must be disruptive both to your your ClinOps and ClinDev people as much as your CMC people. What do you agree with this disparity? And if so, what are you doing about it?
And we do agree with the disparity. One of the challenges, when you're making a breakthrough therapy, you're not making very many batches to identify this wonderful clinical result that you're describing. And so then you're saying 20 patients you've treated, let's file with the EMA, let's file with the FDA. The data is compelling. But at that point, you've made so few batches that when you file with the FDA, they're going to literally say you have no batch experience. You have not done a full validation. You're trying to get approval on a concurrent validation. Both the EMA and the FDA have obviously provisions to do that, but they still will not let you get approval without enough batch experience to show that you've got comparability between the batches, that you have rigor in your analytical methods, they're fully validated, and that you have a process that you can really be comfortable as making the same product over and over again. And what we're seeing is the FDA is putting those places, they're putting the requirements on the companies. They're putting the requirements to have literally a full validation if possible. And I think the idea of concurrent validation, one and done for injection, do one batch for validation and get approval, I think it's getting to be naive right now. I don't think that the FDA or the EMA is going to let that happen.
Stephen, you're totally not helping. You're accelerating products. You're making it worse.
I wouldn't say we're making it worse. Otherwise, we're doing something wrong. Yeah, like this problem's been around for a while. We all know that there's going to be no easy ride if you're on some sort of accelerated path through. There's huge investor pressure, et cetera, et cetera. Then there's huge patient pressure. That's why we all do what we do. We've already sort of covered that. So I think there's a couple of sort of aspects we can attack this. First is we need to invest more in this sort of translational activity. Catapult type of organizations, large academia that are sort of doing this sort of work. They need to have perhaps better entry type of systems here, more understanding of what's needed here. The more we can get that starting point up, then to a level which is sort of adequate, which can then be built upon for the next phase. That's what we're trying to do with the Catapult, whether it's automation, whether or not it's getting some multi sort of parametric analysis done around these products. We might not know what the good looks like, but how can we utilize some platforms in a smarter way to capture that sort of data and then mine it as you're going forward. So I think there needs to be a raising of the bar by that sort of entry level activity, whether it's directly through funders or not. Otherwise, we're constantly in this race. Yeah. Secondly, as we then are going through this accelerated process, the receiving company, should we say, whether or not it's a company organically growing or some sort of partnership, they really need to have, I think, more flexibility and understand what they're getting involved with, and be able to understand that this is not going to be just often a plug and play. There's going to be more activity here. There's going to need more investment in metrology or whatever. Otherwise, this dash to spend as little as you can to get there it's just unsustainable, as we've seen. The third aspect, and I'm not saying this is lowering the bar in any way, shape, or form, but the one thing perhaps we have seen from this COVID pandemic is that if things can be sort of done in parallel, if activities can be looked at in a slightly different way from a CMC regulatory package, etc., then what can we learn from that as an industry to say, look, we might not have everything here, but this is how we can do this on a rolling basis, for example. I think Neil talked about the use of sort of CMC template. I think that would be really beneficial as much as we can. It's great if you've got a platform or some reference point that you can use to say, look, this is what the data package looks like here. So I think there's a couple of different aspects, but ultimately you can't underinvest in your manufacturing process, because if it's intimately involved with your product, you need to understand what you're making. And to me, it's coming back to measurement and just understanding this product more and more in the early days, rather than trying to get away with as sort of little as you can, really, because it's all rather difficult. Yeah. I think we have moved away from that, but it's still more to be done there. And that whole interface between sort of cyber and physical measurement, to me, is still a huge opportunity, and we're not really getting there in terms of how we can use smarter computing to make sense of our data.
You're generating a treasure trove of data over there, Stephen.
Yeah, and obviously we work with a lot of companies, and like others, we focus on what those company needs. And there are certainly, trends and themes that you generally see. And so the more we can template that and give some roadmaps, that's very much what we're doing at the moment, is working with those academic interfaces and micro startups to give them more of a pathway that they can follow quickly and as sort of cost effective as they can. Yeah.
And Rupert, I mean, your corporate, the oral history of the tribe at Thermo is titanic. I mean, you span the globe. You do everything. You must be gathering data of this. But to go back to the question, do you agree with this discordance we see between clinical and CMC in this field?
Yes, I agree with everything that my fellow panelists have said. Sometimes, we have to kind of go back and fix things to move forward, and I think that's happening with a lot of people. What the trend that we are noticing is when people take time to do the preclinical activities in a more thorough and more efficient way, and tighten the processes, make them more standardized, more robust, more reproducible, I think the path to clinical and commercial is a little bit smoother. It's still the concept, I've heard this people say many times, and I'm sure you have, with cell therapy, we're building the plane as we're flying it, right? We're learning things, we're fixing things, in real time. And that will happen for a while, because that's just the nature of the game. But I honestly feel like if we pay more attention to the preclinical development and start implementing some of the principles that we implement in the clinical aspect, keep bringing it backwards and make it more and more standardized, robust, like Stephen mentioned, there's a lot of effort from Thermo for automation, AI, digital, and making everything digital, having all the different instruments connected through like a Delta V system to start monitoring things in real time. All of those kinds of pieces put together will probably give us a very, very robust preclinical process with characterization in process, all of the testing, and moving it into the clinical space and then commercial space will make the road easier, will also allow the reviewers to breathe a little more easily, because sometimes I feel like they're just wondering, hey, what's going to come next? What's different about this? And how do we handle it? Don't think cell therapy is where you can actually create buckets, which is what FDA likes. We're not there yet. At some point, we can start putting things into buckets. And it's what you and Neil mentioned is A template would be good, but I honestly think that there will be renditions of template before we come to a final template. And that's okay too, because it's a beginning of a process that will eventually get standardized.
Yeah, we hope. I mean, Martha, your company's only been around 5 or 6 years. You were founded on research which predated that. But you, in a sense, have had a chance to mold yourself straight into the new paradigm rather than adapt from previous decades of your company's existence, like organizations like Neil's, for example. Do you agree with this discordance, and have you been able to grow yourself around perhaps phase 2 being the new phase 3?
Yes, I mean, I think it's an expectation, and I think that the newer companies entering the cell and gene therapy space, success is defined as having that clinical efficacy that makes you, from a CMC perspective, move fast. So I think the strategy that one needs to take in this situation is to do as much of that planning and additional work setting you up for success, because the expectation is clinical success. And so therefore you want to set up your organization to be ready for the CMC to go along with it. So I think, in a lot of ways as a new company, you benefit because you're already laying down the groundwork in a unique way. You're not overcoming any cultural biases. You're not looking at an established process already that you're breaking out from. You're setting it up de novo, but also with a team of colleagues that generally are well experienced from other industries and other companies. So you have a little bit of the best of both worlds. You have the experience of your colleagues and the perspective of some of the companies that have come before and what they have both accomplished and have run into with roadblocks to help you learn from. Yeah. And panels like this, which are very useful for all of us to learn from each other.
Yeah, no, I agree, and very enjoyable. Well, the hour's gone by very quickly. There's a ton of things, some of which we mentioned yesterday, some of which we didn't, that we just haven't had time to get to. And again, in normal times, we would probably conclude this discussion at the bar afterwards. We'll have to wait till next year for Stephen to buy us all drinks again this time. But I'd like to finish up. I'm going to put you on the spot. From your perspective, I'm looking right now at the FDA list, approved products. Welcome Breyanzi, welcome Tecartus. Very little change there, but another minor milestone, I suppose, in Kite being the first corporation to have 2 products on this board. And I guess the bigger entry there, although from a somewhat troubled path, was Zolgensma from Avexis, which is now part of Novartis. So welcome to those 3 to join the triptych of 2017, but not a whole lot else to be happy about on the commercial side. So 30 seconds from you. Over the next year or 2, is the commercial approval rate going to get better? In the next 12 months, how many are we going to see? I guess so. You can't have too long to talk about. Martha, straight at you. Is it going to get better, and how many?
I mean, I do think it will get better as we learn from the situation as it is. But I think it's probably not an immediate process. So I'd still keep it in the low single digits. Low single digits.
I guess that counts as an answer. Neil.
So I'll go a little more on the limb. I'm going to say about 3 to 5 in the next 1 to 2 years should get approval. I think that people are understanding a bit more what's required, and I see this being a little more productive in the near future.
Stephen?
Yeah, I think there is going to be a COVID lag as well. We can't ignore that.
Which we did ignore in our discussion, but who would...
Yeah, I'm sorry. Yeah, I probably mentioned it, and I shouldn't have. Got to keep away from it. But I'm with Neil. I think it's going to be around that sort of 4 to 5, sort of over the next 18 months.
That's getting better. I used to think it's going to get better. Rupa?
I would also agree, 3 to 5. But I'm also eternally optimistic. So
I was going to say, we're defining ourselves.
Yeah, I'm English, like Stephen, so I'm eternally pessimistic. I'm going to say next 12 months, 3 approvals, 3 rejections. And I guess that is just about getting better. I think the light's on at the end of the tunnel, but we haven't quite turned the corner yet. We have turned the corner on something. This is, I think, the first Zoom meeting I have ever conducted in the last 12 months where I haven't had to tell somebody to get off mute. It's that thing in the bottom left of your screen. You're on mute, hit it. So we're all Zoom pros, and I hope we get to forget those skills really fast. This was a fantastic panel. So Rupert, Stephen, Neil, Martha, my sincere thanks. And with that, we'll conclude the panel discussion.
Thank you, everyone.
Yes.
Thank you.
Fantastic. Thank you.